Tardigrade, Hypsibius dujardini, discovered in 1773 by Johann Goes. These microscopic water bears redefine what survival means. They've endured everything: boiling, freezing, radiation, and yes, outer space. In 2007, European scientists sent them aboard the Photon M3 satellite. They were exposed directly to the vacuum of space for 10 days. When the capsule returned, many woke up and walked away. Completely fine, no oxygen, no pressure, no water, yet alive. Their trick lies in cryptobiosis, a state where they dry up, shut down all metabolism, and wait. Their cells produce glass-like proteins and trehalose sugar that replace water, preserving DNA for decades. Some revived after 30 years in storage. Even their genes borrow from bacteria, making them part animal, part hybrid survivor. But another microbe doesn't just endure radiation, it laughs at it. Deinococcus radiodurans. Found accidentally in 1956 inside irradiated meat cans. This bacterium stunned researchers by surviving doses of radiation 1,000 times beyond lethal for humans. Each of its cells can repair shredded DNA in hours, stitching together its genome like a living puzzle. Nicknamed Conan the Bacterium, it thrives in nuclear reactors, desert soils, and even spacecraft sterilization rooms. In 2015, NASA tested it outside the ISS. 2 years later, it was still alive. Scientists believe its thick cell wall and unique manganese complexes act like molecular armor, neutralizing radiation damage before it spreads. Some call it the blueprint for future Mars colonists. It could help terraform barren worlds. But while Conan thrives on radiation, others prefer the silence of frozen time and salt. Bacillus subtilis spores. When conditions turn deadly, Bacillus subtilis pulls the ultimate disappearing act. It transforms into a spore. Inside this microscopic capsule, DNA and enzymes freeze in place, protected by a thick coat that laughs at vacuum heat and ultraviolet rays. On the International Space Station, spores survived 6 years unshielded in the Expos facility, reviving instantly upon rehydration. That's longer than most spacecraft missions last. Researchers now use them to test contamination risks for other planets. The same genes found in ancient rock samples suggest spores can stay dormant for millions of years, waiting for water or warmth. It's no wonder NASA treats them like biological hitchhikers. Yet while these spores hide to survive, another life form embraces ancient chemistry to live forever in salt Halobacterium salinarum. Trapped in salt crystals for millions of years, these purple-colored microbes still wake up when rehydrated. First found in the 1930s salt mines, they taught scientists that life doesn't need water, it just needs patience. Their pigment, Bacteriorhodopsin, captures light directly, generating energy without photosynthesis. That same molecule could help future solar tech. Some samples revived from 250 million year old halite deposits, still genetically intact. They thrive in hypersaline lakes like the Dead Sea, where no oxygen or plant life exists. Researchers now study them as models for potential Martian microbes, since Mars's salt deposits mirror Earth's ancient brines. Their endurance suggests life on other planets may simply be sleeping, waiting for moisture. But if these bacteria survive by light, another microbe mastered it through ancient oxygen-free evolution. Crococcidiopsis. Cyanobacteria. This microbe may be tiny, but it's rewriting what alive means. Found in deserts, Antarctic rocks, and volcano edges, Crococcidiopsis lives where life should fail. In 2014, scientists exposed it to direct space vacuum on the ISS for 18 months. It survived radiation, freezing, and oxygen loss. Once back, it grew again as if nothing happened. Researchers believe it's one of the oldest oxygen-producing organisms on Earth, dating back 3 billion years, long before plants. It hides under rock surfaces to block UV rays and still manages to photosynthesize with minimal light. Its DNA repair rate is unmatched among cyanobacteria, fixing damage in minutes. NASA studies it as a possible Martian colonist. It could someday seed oxygen on Mars. But from microbes that breathe sunlight, let's move to worms that walked through fire and came back alive. Nematode Caenorhabditis elegans. In 2003, when the Space Shuttle Columbia broke apart during re-entry, nearly everything on board was destroyed. Yet weeks later, scientists found sealed containers that still held living nematodes. These tiny worms had endured freezing, vacuum, and re-entry heat, and they woke up as if they'd just napped. C. elegans was the first animal to have its entire genome mapped, and it shares 60% of its genes with humans. That's why it's used to study how space affects living cells. These worms even reproduce in microgravity. On the International Space Station, they completed full life cycles, proving animals can adapt beyond Earth. What's strange is how calm they stay during stress. Their metabolism slows but never stops. And deep in that resilience, we start seeing life's quiet confidence against extinction. However, some life doesn't just survive, it grows stronger inside spacecraft walls themselves. Aspergillus and Penicillium, fungi. When astronauts complained about strange black spots on the ISS, scientists discovered something eerie. Fungal colonies of Aspergillus and Penicillium were growing on the walls, inside filters, and even on control panels. They weren't hitchhikers, they were thriving. Tests later showed these fungi tolerate 200 times the radiation that kills bacteria and can digest metals and plastics. Their spores survive desiccation, ultraviolet light, and cosmic rays. Some samples from Mir and ISS missions even grew faster in space than on Earth. NASA's now researching them as natural radiation shields, since melanin inside their cells absorbs cosmic rays. The same pigment could someday coat Mars habitats. But among all these space survivors, one tiny bacterium holds the record for being too tough to clean off a spacecraft. Bacillus pumilus, Saphir 032. It started as contamination inside NASA's Jet Propulsion Lab. Tiny, stubborn bacteria that refused to die. Bacillus pumilus, Saphir 032 survived cleaning chemicals, UV radiation, and desiccation stronger than any other strain tested. When sent to the ISS for 18 months, over 60% of its spores revived perfectly, despite full space exposure. Its DNA seems armored with special proteins that repair radiation damage instantly. Scientists now worry these microbes could accidentally seed other planets during missions, a process called forward contamination. Some even suggest Bacillus pumilus could survive the Martian surface for years. Its life so determined that NASA built new sterilization standards because of it. Still, these are Earthborn stowaways. The next survivors live by choice, simple creatures that turn dormancy into a form of immortality. Colpoda cucullus, ciliate protozoan. When life gets harsh, this single-celled organism doesn't panic, it transforms. Colpoda cucullus can curl into a tough shell called a cyst, shutting down every process and waiting for the world to calm down. Scientists dried these cysts for years, froze them, blasted them with radiation, and even exposed them to vacuum chambers mimicking space. Each time they came back to life once water returned. Their outer shell is built like armor, thick with proteins that trap enzymes and DNA safely inside. Some revived after 16 years of dormancy, still able to divide like nothing happened. Therefore, it proves life doesn't need comfort, it only needs patience. But one green survivor didn't wait for rescue. It carried sunlight itself into the void and lived. Spirogyra, algae. This bright green filament of algae looks fragile, but its resilience shocked scientists. During the Expose-E mission on the ISS, Spirogyra samples spent 530 days in open space, blasted by cosmic rays and temperature swings from minus 150 degrees Celsius to plus 120 degrees Celsius. Yet when brought back, they photosynthesized again. Its secret lies in its spiral chloroplasts, the green coils that inspired its name. Those structures protect vital molecules from radiation damage and restart photosynthesis once sunlight returns. Some researchers believe this is what early Earth life looked like: adaptable, simple, yet tough. These algae can also form slime-like mats that hold water and nutrients, protecting smaller microbes underneath. However, One Earth partnership evolved to go even further, combining fungi and algae into something far stranger. Lichen, Xanthoria elegans. Lichen looks simple, a crusty patch on rocks, but it's a miniature alliance of fungi and algae working as one. In 2008, European scientists attached lichen samples to the exterior of the ISS for 18 months. They faced unfiltered sunlight, space vacuum, and extreme cold. When brought back, 70% revived and began growing again. The fungal layer shields the algae from radiation, while the algae keep feeding the fungus through photosynthesis. This teamwork makes them one of the toughest life forms on Earth. Some even thrive in Antarctica's dry valleys, where humidity is below 1%. These survivors may guide future Mars missions since they can grow using Martian soil and sunlight. But one microscopic animal goes beyond survival. It cheats time itself. Rotifer Bedelodea. In 2021, Russian scientists thawed ancient permafrost from Siberia and found living rotifers. They had been frozen for 24,000 years and revived like it was yesterday. These tiny wheel-shaped animals reproduce without males and survive radiation, dehydration, and space-like vacuum. Their trick is a chemical called trehalose, which locks their cells in stasis. When revived, their DNA repairs instantly, as if freshly made. In lab tests, rotifers also survived vacuum exposure for weeks, showing potential to endure actual space travel. They twist, crawl, and start eating seconds after revival, continuing a bloodline that may have begun before the last ice age ended. Therefore, among all space survivors, the rotifer reminds us, life's greatest power is the refusal to end. Thanks for staying till the end. Few ever do. If you love uncovering the strange edges of life and time, subscribe and like. The next world-changing story is already waiting.